All right, so here we'll talk about membrane-bound organelles. And so organelles literally means little organs. And there's many different types, but there's two main categories. We have membrane-bound and non-membrane-bound, depending on if they have a membrane or not. And so the ones that do have a membrane are the endoplasmic reticulum, Golgi apparatus, the lysosomes, peroxisomes, and mitochondria. So the endoplasmic reticulum is going to be a network of membrane-bound tunnels, and so it's going to have an enclosed space, and that is going to be called the cisternae. So if we highlight here, this is the cisternae here. So these are those enclosed spaces. So we have two different types of endoplasmic reticulum. We have the smooth endoplasmic reticulum, and so this means that there is going to be a smooth appearance because this lacks ribosomes, okay? This will be continuous though with the rough endoplasmic reticulum here, which is rough because it's studded with ribosomes. Okay, so that's going to be the big difference between smooth and rough, is rough has ribosomes, smooth does not. However, they have two different functions. The smooth endoplasmic reticulum is going to be important for synthesis, transporting, and storage of lipids like steroid hormones, also metabolism of carbohydrates, and then also detoxification of alcohol, drugs, and different poisons. Big thing here is that because it doesn't have the ribosomes, it has nothing to do with proteins. The rough endoplasmic reticulum has, again, the ribosomes, so it has to do with protein synthesis. That's going to be the main function. So it's going to synthesize or make proteins, it's going to transport proteins and also store them for different things that will be later used by other cells or within the cell. And then these proteins will either like become part of the membrane or become lysosomes, which are going to be our little Pac-Men, or they're going to be secreted within the cell for survival. Next, we have the Golgi apparatus, and the Golgi is going to be very close in proximity to the rough endoplasmic reticulum. All right, so it has this unique structure that kind of has this C shape, and so it's one way to tell the difference between the smooth endoplasmic reticulum and the Golgi apparatus. So this will be in proximity again to the rough ER because of its function. So the function is to receive proteins and different lipids to help modify, store, and package them. So this has stacked cisternae. So just like how we had with the endoplasmic reticulum, the cisternae are going to be these hollow centers that have the edges that are going to start to pinch off. And so this is going to create different vesicles so that we can transport different molecules like proteins and lipids either around the cell or out of the cell. All right, so the process by which we have the packaging and sorting of the proteins. So if we start here in the rough endoplasmic reticulum, we're at the cis end of the Golgi apparatus. So here what we'll have is a vesicle that's pinched off from the rough endoplasmic reticulum, and so this is now called a transport vesicle, which is also going to be membrane bound. All right, then that's going to travel to that cis end of the Golgi apparatus, and now we'll have these contents of the proteins and lipids that are going to be modified as it travels through that Golgi apparatus. So now we're at the trans end here of the Golgi, and we have another budding off of these contents in another transport vesicle. So now there's two paths that these transport vesicles can have. First one is that it is going to be incorporated into the plasma membrane, and then its contents are going to be spewed out of the cell. All right, so this could be like something like hormones that are going to be produced within the brain and then released, say, within the kidneys. Another option is for that transport vesicle to stay within the cell, and what it will become is a lysosome. And so a lysosome is going to have these different enzymes where it's going to be our little Pac-Man and help degrade materials, and this will also stay as a membrane-bound organelle. So the lysosomes, again, which are going to be the transport vesicles that stay within the cell that is produced by the Golgi apparatus, is going to have different enzymes that we have in here. And so in this picture, here's an example of different enzymes. And these are important because they help to digest and remove different waste products and get rid of damaged organelles within the cell. And so this is called autophagy. So when the cell is dying, what it does is it releases these lysosomal enzymes, and that helps to digest that cell, and so we have death of the cell. And that's called autolysis. Example that this happens all the time within red blood cells, because they circulate within the body for about 120 days, and then those contents are going to be expelled out and then degrade that cell so that we don't have waste products and dead cells circulating within our bloodstream, for example. Another membrane-bound organelle that's very small in size are peroxisomes, and so these also are vesicles that are formed by pinching off from the rough endoplasmic reticulum, and their purpose is going to be to detoxify different harmful substances. And so what it does is it goes from hydrogen peroxide and makes it into water. This is very important because of metabolism. We'll see this these peroxisomes being active then. And also big use of these peroxisomes is for breaking down fatty acid molecules so that they're not harmful and that our body can absorb that substance later and use it as nutrients. All right, next we have the mitochondria, and mitochondria has this bean shape to it in terms of the organelle structure and appearance. And the mitochondria is a little bit unique because it has an inner membrane and an outer membrane. So this inner membrane, which is, I'm trying to highlight right there for you. All right, so this is going to be the cristae. All right, and what this does is it helps to increase surface area because the function of the cristae is to produce ATP, and ATP is what we use for our energy source. And then what is in between the cristae, so this darker spot here, this is going to be the matrix. And so the matrix is important because it has a lot of chemicals that are needed for the cristae in order for us to generate ATP. And so certain cells will have more mitochondria, so like the heart, skeletal muscle, than others, because these are going to be main sources of energy that are needed for our body. All right, and then the most important organelle for our cells is going to be the nucleus. So the nucleus is going to be the control center for all of our cellular activity, and there's three different components that make up the nucleus. The first is going to be the nuclear envelope. All right, so the nuclear envelope, which I'm highlighting here, has a double membrane, so similar to that of a mitochondria. All right, now here though, the nuclear envelope is important because it's going to control the entry and exit of molecules from the nucleus and the cytoplasm. So the outer membrane is going to be continuous with the endoplasmic reticulum, so like what we have seen here. And the nuclear pores, which are going to be the little holes that we find within the nuclear envelope, has these different channels that allow different molecules in and out of the nucleus. All right, the second feature that we have is the nucleolus. And this is going to be a dark staining body that's found within the nucleus. Here what we have is that it's composed of different RNA, enzymes, and different proteins that's going to make up the components of the small and large units of the different ribosomes. So very important for then the function of proteins, because ribosomes, their function is to make proteins for our cells. All right, so last but not least, what we have is DNA, chromatin, and chromosomes that also are composed of the nucleus. So the nucleus holds the DNA, or deoxyribonucleic acid, which is a molecule that has all of our genetic material. So when the cell is not dividing, the nuclear DNA is wound into these fine filaments called chromatin right here. There we go, chromatin, okay? And so this helps to increase the surface area, again, so that we can have a lot of genetic information. So during cell division, though, then we have the chromatin is going to coil tightly, and so what we'll have are chromosomes.